Hi friends, today we are going to see the past year NEET questions for the chapter Respiration in Plants, and today in this video we will cover part 4, which is the last part for this chapter. Now let's begin with the first question here, which was asked in the year 1995. Okay, which one of the following is essential for the conversion of pyruvic acid into acetyl-CoA? So you're given 4 options here, so we'll go one option by option, right? So what is the full form of LAA? It is called as thymine pyrophosphate, that is for TPP. Now what is LAA? See, in this process we have lipoic acid. So LAA is a lipoic acid, TPP stands for thymine pyrophosphate, and NAD, you all know it's nicotinamide adenine diphosphate. Now which one of the following is essential for the conversion? If you see, all these are required. Okay, during aerobic oxidation of pyruvic acid, first of all there is activation of pyruvic acid, in which NAD and coenzymes react with pyruvic acid and oxidative decarboxylation takes place, which leads to the formation of acetyl-CoA. So initially NAD is very important, but apart from that they have mentioned coenzymes. These coenzymes are the ones that are here. They are all coenzymes which help in the formation of acetyl-CoA from pyruvic acid. So they are all essential for this reaction. Therefore, the correct answer to this question is option D, all of these. Next question here, which was asked in the year 1994. Positions like cyanate inhibit Na plus efflux and K plus influx during cellular transport. This inhibitory effect is reversed by an injection of ATP. What does this demonstrate? Okay. So what do you mean by efflux and influx? Efflux is the outward and influx is the inward. So active transport is a uphill movement of particles against your concentration gradient. So this we know, right? What is active transport? It is the transport of uphill movement. It's a uphill movement transport of particles which is against the concentration gradient. Energy is utilized in this form of energy. So we all know for active transport we require ATP, right? We require the usage of ATP. Energy is utilized in the form of energy. ATP is utilized in the form of energy. Now, cyanate inhibits soluble absorption by lowering the respiration rate, leading to the formation of less ATP. Therefore, addition of ATP will facilitate active transport. So this is used for what? Option A is the right answer. ATP is the carrier portion in the transport system. See, when you add ATP, it will facilitate active transport. Therefore, you are adding ATP. So, ATP is the carrier portion in the transport system. Therefore, the correct answer to this question is option A. Okay? Okay, now let's move on to the next question here. End product of citric acid or Krebs cycle. Very easy answer, right? Very easy question for you. So we all know a Krebs cycle, which is also called as citric acid cycle or Tca cycle. Anything. You have so many names for Tca cycle. Now this cycle, if you see, happens in the aerobic conditions. We all know glycolysis is just the opposite. It might or might not require the presence of oxygen. Even if there is presence of oxygen, it can act. Even if there is no presence of oxygen, the cycle can move on. Now, apart from that, the Tca cycle can be described as a tricarboxylic acid cycle, and it is a series of chemical reactions used by all aerobic organisms. Apart from that, what you can say is, it is the stored energy through the oxidation of acetyl-CoA derived from the macromolecules that you see. By the macromolecules. What are the macromolecules involved here? Carbohydrates. What next? Proteins and fats, right? Proteins and fats. And we know any cycle will release 3 products. It will have an acid, it will have carbon dioxide and water. So the end product of the citric acid or the Krebs cycle is carbon dioxide plus water. The 2 molecules of pyruvate are completely degraded in the Krebs cycle to form 2 molecules of ATP, 8NADH2, 2FADH2, carbon dioxide and water. So if you look at the options given here, you don't have anything which talks about NADH or FADH. So The end products which come among the cycle are carbon dioxide and water. So that is there in the option. So you will mark option D as the answer. Okay, so this is the TCA cycle that I've given you here. So you can see here, right, you can see the TCA cycle here. So this is a TCA cycle. I've just given you a picture so that you can see the cycle and go through. Therefore, the correct answer is option D. Now, next move on to the next question here, which was asked in the year 1992. Oxidative phosphorylation is a production of dash. Again, this is an easy question for you. What is oxidative phosphorylation? Addition of phosphate. We already discussed about it in the earlier parts where we saw it is the addition of phosphate group. Now, oxidative phosphorylation is the synthesis of the energy-rich ATP. We all know that, right? With the help of the energy liberated during oxidation of reduced coenzymes produced in the respiration. So it is the production of NADPH, or is it ATP or ATP in respiration? Of course, it is not in respiration. It is a process which takes place as an NADPH in photosynthesis. Therefore, the correct answer to this question is option B. Now, what is the full form of NADPH? Did you ever think about that? NADPH can be described, or it is abbreviated as nicotinamide adenine dinucleotide phosphate. It's an older notion. Right, this is a cofactor used in anabolic reactions. Okay, it is a cofactor that is used in anabolic reactions. Apart from that, you also have Calvin's. This is an important process in Calvin cycle. You have so many other cycle which plays a very important role, like Krebs cycle, glycolysis. So it is a part of that. The molecular formula of this is C21 H29 N7 O17 and P3. Therefore, the correct answer to this question is NADPH. They are talking about the oxidative phosphorylation. Of course, it is a synthesis of energy-rich ATP molecules. But still, what comes as end product, or what helps in this is NADPH. Okay. Next question here, which was asked in the year 1992. Terminal cytochrome of respiratory chain, which denotes electron to oxygen. Electrons to oxygen is dash. What do you mean by terminal cytochrome? How can you define that? Terminal means it is present in the end, right? We all agree with that. What do you mean by cytochrome now? See, terminal cytochrome, the EST, which is called as the EST system, contains various electron carriers such as cytochromes. We all agree with that, right? Now, what do you mean by this terminal cytochrome of respiratory part? See, what does ETS stand for now? ETS stands for electron transport system. Okay, now here the electron transport system is formed by multiple complexes at different energy levels, which in turn are formed by certain coenzymes and cytochromes. Okay. The coenzymes are the they are the proton carriers, and cytochromes are the electron carriers. So the activity is actually separated, or it is parted between two. Cytochromes will carry electrons, okay, and coenzymes will carry protons. The terminal complex 4 is formed by the it is formed by A, and it is cytochrome, that is cytochrome A and cytochrome A3. Electron from cytochrome A3 will finally get accepted, or it will get accepted by the oxygen. Therefore, the correct answer is cytochrome A3, which is option D. Okay. Terminal cytochrome is the cytochrome A3. It possesses 2 copper centers. It helps in the transfer of electrons to the oxygen. Therefore, the correct answer is option D. Always remember, ETS will have 2 systems. One is for protons and one is for electrons. Protons are carried with the help of coenzymes, while electrons are carried with the help of cytochromes. Okay. Next question here. In germinating seeds, fatty acids are degraded exclusively by dash. You have 4 options here. Obviously, it is not mitochondria, so it should either be perioxysomes or glyoxysomes. Proplastids, plastids are related with pigments, so that is also not the answer. Here they are talking about degradation. Therefore, the correct answer goes to glyoxysomes. In germinating seeds, fatty acids are degraded exclusively by glyoxysomes. Now, what do you mean by glyoxysomes? What is a oxysome, first of all? See, You have perioxysome, already we know what is it, right? Now glyoxysome can be described, or it can be defined as a specialized perioxysomes that is found exclusively in plants and also sometimes in the fungi. Okay, seeds that contain fats. I'm talking about the seeds that contain fats or oils, especially the seeds include corn, soya bean, sunflower, peanut and pumpkin. These are seeds which are highly rich in fats. So what happens is they have to be degraded in order to germinate. So glyoxysomes is the one which will degrade these fatty acids, and they are oxidized with the help of acetyl-CoA by the perioxysome beta oxidation enzymes, which is a glyoxysomes. Therefore, the correct answer is option B. Glyoxylate cycle occurs in tissues rich in fats such as those of germinating seeds. The enzymes involved in the cycle, which have not been found in mammals, are contained in the organelles of glyoxysomes. Okay. Next question here. Respiratory quotient. Respiratory quotient RQ for fatty acid is dash. Easy question. This is a standard question and it's a fact. You cannot change it anyway. Respirometer is an instrument for measuring the respiratory quotient. Okay, we all know that. Now RQ stands for respiratory quotient and it is defined as the ratio of molecules of carbon dioxide. So what is it called as? The ratio of carbon dioxide given out to the number of oxygen molecules. It is denoting to the oxygen molecules taken during the transpiration or respiration. It is a value for fatty acids and it is always less than unity. Therefore, the correct answer will be option B. This is a standard value, you can't change it, so you have to remember or you have to by heart it. Remember, the respiratory quotient for fatty acids is always less than the unity. Option B is the right answer. Next question here. Apparatus to measure the rate of respiration. Very easy question. It's a respirometer. So this is the respirometer that you can see a diagram here, right? Respirometer is an instrument for measuring respiratory coefficient or a quotient as well as the rate of respiration. The correct answer is respirometer. Next question here. Now this respiratory quotient is a ratio of dash. Just now we saw in the earlier question, it is the amount of carbon dioxide produced to oxygen consumed, right? Therefore, the correct answer to this question is option B. Ratio of the volume of carbon dioxide that is produced to the volume of oxygen consumed in respiration over a period of time is called as respiratory quotient. We already discussed a lot about it in the earlier question where we spoke what is respiratory quotient, and it is always less than unity, right? So this question is once again easy for you to answer. Next, so the answer for this question is option B. Next question. Now this RQ, which is called as respiratory quotient, is dash. Again, there is, see, all these four questions that we saw in the last are all resembling the respiratory quotient. It is something that is measured with the help of a respirometer, and here if you see, it is the amount of carbon dioxide that is given to the oxygen that is overall consumed. So what will be the answer? Option C will be the answer. So here you have the denotation here. Respiratory quotient is equal to volume of carbon dioxide evolved by volume of oxygen absorbed. Therefore, the correct answer will be option C. Right? The current estimate is that about 30 molecules of ATP are produced when glucose is completely oxidized to carbon dioxide. So always remember this. You should always you should have this in mind. You might get questions from this. This question which was asked in the year 1988. Okay. So if you see here, the instrument that is used to measure the respiratory quotient is called as respirometer. We already saw how the apparatus looks. You even get it in online, those respirometers. You get it in online as instruments, or you get it for play purpose. You have even respirometers for that. So you can go have a glance of what it is. Therefore, the correct answer to this question is option C. So with this, we complete the part 4 of respiration in plants. This seemed to be very easy for you because we just ended up with this chapter. So we will all meet in the next new chapter. Thank you for watching my video.